Okay, hi Ben.
Hey, how are you, Norman?
Good.
Good to see you again. So, what's new? No, I was going to bring up the study at the Antarctica. I guess that was an interesting one. I think I sent you it, right?
Yes, and there is a similar study that was done on a Scottish island. I sent you a link to a. I didn't find the original paper, but it was quoted by both papers are quoted by this Carl Hennehan and Tom Jefferson in their blog. Follow the evidence on Substack, and this is very, very interesting. And of course, you cannot say that there was no contagion, because definitely these are, you can find the same virus in many different people. But the thing is that it's very difficult to say how we are infected and how it looks, for one reason. The diagnosis of colds is very complicated, and if you read the CDC EPIC papers from 2015, and there are two important take-home messages. One is that 62% of cases of severe colds of pneumonia, the best labs in the world could not determine what is the etiology. That means what was the cause. So it's obvious that diagnosing colds or respiratory infections is very, very difficult for many reasons. One reason is that a simple diagnosis by taking a swab from the nasopharyngeal, that means from the nose and the throat, and from deep throat, which is called oropharyngeal swabs, it's not really good, because what you are testing the mucus. That means you are testing our air filter. So something that came in, if you are using PCR, you can find one molecule of something on a swab. So it's, if you look at the EPIC papers, it's clearly stated that from swabs you cannot diagnose a respiratory infection. You can diagnose it, and I was doing that from, of course, I was not taking the samples from patients. This is done by doctors or hospital staff. The best diagnostic sample, which is also very difficult to get, and it requires, of course, the consent from the patient, is to do so-called bronchoalveolar lavage, or BAL to make it simple, which means they put in a tube in the lungs of a patient and then wash out whatever is there at the end. That means from the alveoli. So this sample is very difficult to get, and in the EPIC study, where they took samples from about 2,500 patients, that's the apex. You cannot do anything better than that. And all other studies that were done in the world, even the Chinese study where they found this Wuhan virus, Wuhan human 1 virus, known after so-called renaming by Drosten and his accomplices as a SARS virus, although it's not really similar to the SARS virus, because 80% sequence similarity in evolution means nothing. So they also found another thing in this study, that the most common virus in severe lung infection is not the flu virus, but it's the rhinovirus. As you notice, there is nothing about the rhinoviruses in the media, nothing. You never heard of rhinoviruses. They are, of course, scaring people with the flu because a lot of people remember wrongly that the flu virus killed several million people during the end of the First World War.
1919, yeah.
That's a lie. That's a lie, because there is a paper published in 2008 that all pulmonologists should know that the people were killed by bacterial complications after whatever was this respiratory infection. Nobody knows it was a flu virus or any virus or many different viruses. So people are remember, and they are scared of flu. They take the flu vaccine, although the flu virus doesn't cause a lot of respiratory infections. So this is kind of a big lie that people are scared into taking the flu vaccine. The flu vaccine, as we know, doesn't work.
Yeah, well, let's get to the flu vaccine, but so I think what you said is that with the testing of the flu bronchial lavage fluid, or like the swabs in those, you said it's essentially like a testing like an air filter. If I was to take my air filter at home and I would look what kind of pollen or what kind of things in there, and so you would get a lot of false positives because it could be just a single virus that's being stuck in there, but it doesn't mean it has entered your body and it has infected me or someone, right? So it could be just there. And then, I mean, you also have the fact that there's a lot of other host organisms there, and also host genetic material as well, right? So you could, like, I mean, that's probably where you could help me out to understand. But couldn't you, if you cranked up the PCR test like really, really high, couldn't you just also kind of get a positive on, say, bacteria or human genetic material maybe, or something? Do you think that's possible? Like, it kind of goes into the direction of like the chart I've sent you. I don't know if we can share the screen on here, but I guess we can.
Yeah, you can share it in Skype, but you see, the thing is that it's worse than you think. That means if you have a complex sample, and definitely a swab, as you mentioned, can contain a lot of different, in the mucus, you can have host DNA, you can have human DNA and RNA, because then you can have all kinds of other organisms. You can have 1 million of different organisms. So you run a PCR, but this PCR that they were doing during the COVID scare and the COVID scam was this so-called reverse transcriptase quantitative PCR. What do you see as a result? You see a fluorescence plotted against
a line
cycle.
You see a line, yes, and that line, it kind of seems, maybe it's always kind of going up at some point, but the cutoff is where the difference is, right? So
but the thing is that you don't really see the results. You see only the fluorescence, which doesn't mean much, because if you would take the sample and analyze at all these points, you take a bit of the sample and look at it at different points, you would see that what accumulates, even if you have, there was no target, no real virus that the primers were designed for. You see the accumulation of pieces of DNA of various lengths. This is called a smear. Now, this, because at some point, if there is you have a very high concentration of primers, and they can attach to anything, because you go through the cycles of denaturing the nucleic acids, then aligning the nucleic acids, because what you RT-qPCR means you use the reverse transcriptase enzyme to convert the RNA into DNA, because PCR is really, geared toward DNA. You cannot do PCR directly on RNA. So you RT, you reverse transcriptase, you convert the RNA, the RNA into DNA. So
cDNA, right? Complementary. Yeah.
Complementary DNA. Yeah. So the PCR has, let's say, 3 steps. There is, you can also do isothermal PCR, but we are not talking about that, because in the regular diagnosis that was done for this fake pandemic was this classical PCR. That means you heat up the sample to 90-some degrees to denature the DNA, then you bring down the temperature, let's say, to 55 degrees to anneal the primers to the template, and then you raise the temperature, let's say, to 72 degrees, something like this, to extend the primers. Yeah? Mm-hmm. And in this way, you produce more template, and it goes as a a chain reaction. You get more and more of the product. Yeah? And then, of course, that's qPCR. That means that means in the reaction you release a quencher of a chromophore, and you produce a fluorescent signal. This is how it's done. So you don't see
the signal in the.
But you don't see what's really happened in the sample. And if you have mispriming, and you see this smear, a collection of all kind of. So if you have a complex sample in which you know that there is no virus, if you run to 35 cycles or 40 cycles, you will see a signal. But you see a smear if you analyze what you have in the sample. That means you can get a signal without a target. So unfortunately,
what do you mean by smear then? At this point, you mean like the curve of the signal?
No. The smear is if you would run on a gel, the reaction. Okay. That means you see pieces of different size. Theoretically, you should see pieces of a piece of one size, delimited by the primers. But the qPCR is a bit different. But whatever, it means that what you see, you would see even if there is no target. If you run many cycles. So many cycles means that you can, the primers do not have to fit very well. But if they will suddenly produce something, then it gets amplified, and you get a signal.
So when you actually use no primers, basically, when you use no specific amplification, just amplify everything essentially, then you get a smear, like you said, and it's sort of like all over the place, right? Like you get all sorts of different lengths.
Yeah.
And if you use amplification with a specific primers, you would only get certain areas which are present.
If you have a target.
Yeah.
So you have to have primers to push.
And that's on the gel, and then they centrifuge it, I guess, right? And it sort of like moves through the gel, and
a gel separates agarose gel or acrylamide gel. Acrylamide gel separates fragments by size. Okay?
Yeah, so
you don't need a centrifuge. You just put it on a gel, run,
and then we'll just move through it like
then
slowly or something.
Then stain it, and you see something like that. If you are using this RT-qPCR, you just see the signal, which doesn't mean that the target was there. Because what you see are molecules where the quencher is removed from the chromophore. Okay, and this can happen without any target. Because you add primers, and you have one of the primers or both have, or you can use multiplex, you can use more primers, but different quenchers, different chromophores. So you produce a signal for the fluorescent signal, but there is nothing there.
Yeah. So like the signal basically is another abstraction step, and it may produce also like a false signal essentially, or like a because
and this is explained.
And so
this is explained in the EPIC papers. It said that if you do PCR on swabs, this is not valid diagnosis. Period.
Yeah. So the agarose gel, right? That's a good keyword. So I'm not sure. Are you aware of? I think it's been a long time since I looked for it, but I couldn't find actually a paper or any description where they basically extracted the RNA of SARS-CoV-2 and put it on like a gel and found exactly the length of, say, 30,000, 29 kilobyte or 30 kilobyte.
No, this is not really easy to produce intact RNA.
Mm-hmm. Yes, it will through the process. It will automatically split up already.
Yes. RNA is very difficult to get in a pure state, and then such a long molecule is very difficult to amplify, to convert into DNA with this reverse transcriptase. So you cannot really get the whole virus like a RNA virus as DNA. So what you do, you know, some viruses were in the past, coronaviruses have a long history from the 60s. They were later sequenced and assembled by simply looking at overlapping pieces. So you can use this sequence of adenoviruses, you can feed them into your sequencer, and you can then overlap pieces, and you can more or less create a sequence of the virus. But you know, this is not really important, because you know, this virus assembled from pieces, even on a you know, master sequence of something, will be okay. It's not really important to have a very detailed sequence of a virus if you are just talking about a virus that you sequence for fun. But we are talking about a virus used for pandemic, and this is again not important. The virus is not important. The important is the diagnostic procedure.
Yeah, I agree. Yes.
Yeah, although probably hundreds of thousands of viruses were of the Wuhan, I prefer to call them as they were called by the original people, because remember, Wuhan human 1.
Yes.
WH1. Calling them SARS by Drosten and his group of people that said, okay, we are the committee. We can call. We can.
Yeah, I mean, it's like I see that basically the issue is that, like, so the technology that's been used may have some merit, let's just say it like that, or it seems in certain certain applications seems very valid. However, how it's been used in the pandemic, it seems like it's a gross misuse of it, like, because they've basically just used the swabs on everyone and then just amplify the signal, and like it's such a high abstraction level that we don't even know what it's amplifying at this point anymore. Right, that's basically the issue with it. Let me try to share the screen now. I want to see. Can you see it, or? Yeah,
I can see it.
Okay, I can't see it on the video. I don't know if it's gonna come through, but yeah. So I mean, this goes to the question of the primers and the target, right? So like here we have. So I took this, I think, from these are the primers from Drosten's PCR test, like down here, the source, and then so let's just look up here. We get a primer. This is the primer here. It's like 22 nucleotides long, and here's a match. I think that match, if I remember correctly, that one was on, I think, like a human genome or part of a human. Part of the human genome, maybe, or something. So I've highlighted here the red areas, which is essentially a mismatch. So you see the X could be anything at the ends, and then here is an error, like with the CG. But it's very close. We get like 18 out of 22. We get matches, so 82%. And then for the other one, the other possibility, you have the primer here in yellow again, and then you get maybe two overlapping ones where you it perfectly matches. So this is just an idea, like, because I guess I don't have too much knowledge on how the PCR really works and in the detail level, and maybe nobody really has, but right, I don't know. But maybe, do you know if these scenarios are possible? Like, do we really know what the primers react with in a sample?
Yeah, but this is just checked with human DNA. But you see, let's say let us look at this first pair. The thing is that depending on the annealing temperature that you get, you force that, for instance, 17, let's say here are 18, that 17 or 22, 17 of the 22 of the 22 base pairs should match. You can tweak the temperature, and you can select how many bases should match. However, these are bases in a contiguous sequence. It's not 5 here, 5 here, 5 here, because if it's broken, then the annealing won't work. And then at the end, on the right side, where is the 3' end, it must be a 100% match. Of course, here in the first is XX. Yeah, so anything, they say that there are multiple sequences in the human genome. But you see, apart from that,
so we wouldn't get a signal at this point. Like, if it's not 100% the enzyme or wouldn't move along, and then it wouldn't release the fluorescent, right?
Depending on the temperature.
Okay.
Because
so if you heat it up enough, that's where it sort of like breaks down, and like you would still get it would still kind of move through, and it would at the end it releases the fluorescent signal, right? That's how it works.
If you have a problem with sequence that is, partial match, and you do not discriminate by using different temperatures, then you have a problem. So the thing is that in the PCR, because in the reverse transcriptase it's done at low temperature, so it's completely different. We are not talking about the primers used for the reverse transcriptase, but for the PCR, which requires higher temperatures. Yeah? And then you can fine tune the temperature to have just one product. But of course, the thing is that if you take a swab from someone, it's something completely different than the company tested, because you walk around, you get all kinds of viruses, bacteria, pollen on your mucus, and then it's tested with the primer, so you don't know what you have. It may be a coronavirus from an earthworm or a cockroach. You don't know.
Right. I see also like another inherent problem of this, which is that if like you take a swab from a person that say only would have one virus, and you are able to fetch it in your sample, you get that one virus on it, but because it's just one, there's very low amounts of the genetic material to amplify. So you would need probably a very high temperature to find that piece, right? At the same time, with the high temperature, with the many cycles you have to run it, wouldn't you also increase the likelihood of other matches, other bad?
Yeah, because you have such a complex sample that you have to be very careful and really test the sample, without the virus and with the virus. So this is not doable. That's why the EPIC papers then say you cannot use swabs for diagnostics, period.
Yeah.
And they were used. That means nobody read those papers. These are papers that were also released as a news item.
Well, I mean, they're doing it again now with bird flu. I guess that's what they're doing, right? When they go into there was an outbreak of hand-mouth foot disease in Germany recently, maybe a month ago. They found, I think there was a couple of water buffaloes that actually they're not home in Germany in the first place. They come from somewhere in Asia, I believe.
Yeah.
So they're not native to Germany, and so I think one or two maybe died in the winter. So then they tested them, and they found the hand-mouth-foot disease sequence. And then they started testing everyone else. I mean, all the other animals, and apparently they found some, so they got all killed. So I mean, they're using the same technique, right? And the same with the bird flu in the US now, where they're testing, I think, the chickens and stuff like that. So isn't it exactly the same procedure?
You know, the problem is with the bird flu. Of course, there are some theories that this was the last, because you know the price of eggs went up 300%. I have to take.
No problem.
Call me in about an hour. Thank you. Yeah. So the thing is that with the bird flu, they say that there was a Chinese origin lady at the USDA, the United States Department of Agriculture, who decided to kill millions of chicken because of the bird flu. But the bird flu, they say that it can infect people. Some years ago, maybe 2 years ago, they said that bird flu can pass through from pigeons to cats and then infect people. But people in the West were not really that happy to bring their cats to be put, to be killed.
Yeah, that's the whole problem, right? Like it doesn't seem like we have a really good sampling, because that's what if I were to like really go down methodologically to try to approach this, like what does this testing really mean? What I would start with, I would establish a baseline, right? I would have to go out into the population, get maybe 1,000 people to sign up randomly and test them and try to look for all sorts of viruses, sequences, whatever. So, I mean, that doesn't look like it's been done. The same thing with the tests, like for the bird flu, like I think there was one case in Texas where they said like, oh, it jumped from the bird to the human, but they haven't.
So what?
So what? Yeah, sure, but like, I mean, that the problem isn't a problem that they haven't, to my knowledge at least, they haven't gone out with the test and looked at 1,000 healthy people, 1,000 people that are already in the hospitals for something else. So it's sort of like not really validated. It's not. No,
but Ben, you should know it. U.S. mortality. If people are dying,
yes.
Now, if more people are dying than usual, then you should test. Because if you go and test, you saw during this Wuhan virus pandemic, people are not sick, are found to be infected, they are put in the hospital and killed at the hospital.
No, I agree.
Do you prevent a disease by condemning people to death who are not infected? This is so crazy.
It's totally crazy. I'm totally on board with that. I think they should have never started mass testing, never tested healthy people, because it just creates a lot of fear, a lot of panic in the population. Again, going back, because I mean, it does make sense under the assumption that the test is really finding the thing that you are looking for. But I think as we have worked out, we cannot be 100% sure. We can't be really sure that it's really always finding just the virus that we're looking for.
Yeah, but
the problem was that they sold it as novel, right? So, I mean, that's the whole other thing that because that's what people were scared. Like they were saying like the CFR is like 8% or 3%. So of course, if someone tells me, hey, there's something going around with a 3% mortality rate, or 5%. I mean, at the beginning, when I had no idea of what's going on in March 2020, we were all scared, because people thought like, okay, out of 100, 3 people will die or 5 people will die. That's crazy. But I think once you realize it's all based on false statistics, in my opinion.
Yeah, but look at 2020, 2021. They were finding the Wuhan virus in all animals they were testing, and this was presented by scientists. This was presented by scientists to the governments to get more money of testing wildlife. But now look, the coronaviruses are species specific. So how come a human virus suddenly started to infect everything? They were killing the mink in mink farms in Denmark and in Poland, as I know, claiming that this virus can be spread by those animals. So it took the Wuhan lab to produce a virus. No, yes or no, maybe. And then this virus can infect all animals and then infect people. And only after some time, some scientists probably prevented in scientific journals, and they said that no, these viruses cannot infect other animals, and these are just PCR errors. But it never made it to the public. Also, New York Times once published an article that your test was positive, but maybe it was not. And this was in summer of the 2020. But still, this that maybe it was not disappeared, and they wanted people to be positive on that test to kill them in the hospital.
It was a complete, how do you say, panic or craziness?
Scam. This was a scam.
Essentially, yes, probably. Well, the problem is that they are still using the same techniques right now. Bird flu, and I mean, that's the only thing they can do. And like, for example, they have also, to my knowledge, they have not done any blind testing. That's what I would do if I was in the government, in the CDC, or let's just say, let's just get some samples of people and let's test them, but let's not tell them what they actually have. Like, I mean, it's sort of like, of course, I don't know if that's ethically possible, but because only if you not tell people what we found in the tests, we can really establish if the results of the testing have any causality to the outcome, right? I mean, if we tell people, hey, you have a virus with a 5% mortality rate, like that does something to people. So
yeah, but look, many universities all over and laboratories all over the world, they get money to test wastewater, and they publish graphs of appearance of the Wuhan virus in wastewater. And in 2020, a paper appeared, which is the, let's say, review of the literature on finding viruses in wastewater. Of course, they are viruses that are very environmentally sound, and then they are protected. For instance, like, the poliovirus. You get polio from the water, contaminated water and food. Yeah. So, of course, in recent years, when water and food doesn't contain fishes as much as it was in the past, people don't get those diseases. The same cholera and several other diseases. So, finding viruses that can pass through the alimentary tract and survive in wastewater makes sense. But the enveloped viruses, which are very labile because they have just a membrane, like membrane of our cells on the outside, like the flu virus or coronavirus, they are not going to pass through our intestine intact. And then RNA is very, of course, unstable. So if the capsid of the virus would open, RNA would be gone very quickly. But people are finding that, and definitely nobody talked to the regulators or to committees that give, money to scientists, that this makes no sense. And in this review article, of course, this is published in 2020, so it says, yes, you can find, they found 3 papers that you can find coronaviruses, even SARS-1 in wastewater. But there is no other envelope virus that they say people were looking for during the last, 50 years. Yeah? So PCR is around for 40 years. So they never looked for the flu virus. Why? Because you cannot find it. So
in wastewater,
you cannot find it because it's an envelope virus. If you know it passes through your alimentary tract and into the wastewater, you'll be gone, long gone. You cannot find it. And in this paper, they say, oh yes, you can find the coronavirus, but they don't say it about, they mention a lot of different viruses, but not the flu virus, because of course you cannot find the flu virus there, but flu virus and coronavirus have basically the same outside structure. So
you're saying they should also not find the coronavirus?
Of course, this is fake, but there is so much fake science. Take global warming, there is so much fake science. Most science about the coronavirus is fake, because how the coronavirus could replace all other viruses that were causing colds? That's impossible. There are almost 1,000 such viruses.
Yeah, I mean, exactly. So like, I've tweeted that a couple days or months ago. Like, it's been 5 years now since the novel virus has gone around the world, but we still don't know where the flu went. Like the common flu disappeared from the WHO charts in 2020, 21 until 2022, almost 2 years, I believe. And nobody has come up with a proper. I mean, I think the most common thing is like, oh, it's viral interference or something. But
no,
I mean, how
doesn't happen?
You would have to show how that happens, because I can find papers where they have both, where they have co-infection or something like that. I know a paper that I looked up a long time ago that said that coronavirus helps the influenza virus actually or something. So they can definitely coexist in the same isolate or sample, or so it doesn't really make sense to me. But yeah, I mean, it's crazy. It's like, it almost feels like there's a lot of pseudoscience going on, for sure. I mean, it's because it's very complex, the whole topic, and but yeah.
Yeah, but the government will give money for anything stupid, so
that's why I was surprised when I asked the CDC if they have. I asked the CDC and the RKI of Germany, the German equivalent of the CDC, if they went in and kind of what I call validated the sequence, which is like to really go in and poke some holes into the process. Because like, as I explained, I think in our first session, the Wu et al. Paper, they basically just used the next generation sequencing to find this novel sequence, right? So they never went in and did the additional steps to validate if this is truly the sequence of the virus, which you would expect. Because I mean, now I think I found like two studies that have Sanger sequenced it. So there are maybe Sanger sequencing of it, but it's very rare actually. Yeah.
But you see, the first thing was, we talked about this some minutes ago, SARS-CoV-2. And I say, no, it should be called Wuhan virus. And they say, come on. It was named by a committee for naming viruses. I said, look, did they forget about the MERS virus? Why didn't they rename the MERS virus SARS?
Exactly. Yeah.
And people say, no, it was a different virus. No, it caused SARS. SARS is severe acute respiratory syndrome. It's the name of the disease. The MERS virus causes SARS. So why didn't they rename it? They renamed it because they were in cahoots with Pfizer, who wanted a good name to sell the vaccine for. So that's it. No, there was the MERS virus only.
Yeah, in the south.
It's not really transmitted from person to person. No? So they didn't want to talk about this. But why didn't they rename it?
Well, I think they probably renamed it because they officially, I mean, in the paper it says that because it sort of causes the same, it's thought to cause the same symptoms, and also like it has a high similarity to SARS-1. But as you said, I think it was like 80% or 89% or something. I think that's similar to what a rat is to a human or something like that.
No,
So,
less.
less
But
than
then
that.
MERS is only 50% similar to the Wuhan virus. Sequence similarity.
Well, and both of those actually disappeared, right? So were outbreaks, local outbreaks somewhat, like MERS was in the Middle East, I believe. I don't know where exactly it was. And then SARS-1 was also, I think, in Taiwan and maybe in
United
China
States,
as well.
Canada had a lot of deaths.
Okay, SARS-1, I think, too, yeah. But it was more punctual. It was more like where it seemed almost like wherever they tested.
Nobody was tested with PCR.
Yeah.
People were tested just, at the airports with the temperature.
Oh, okay. I didn't know that.
No
so they
PCR.
didn't run the sequence. Okay.
PCR was around, but who cared about PCR? They were finding people.
So then it means they actually identified people based on the clinical symptoms at that point, and they assumed people that had come from the region and had a fever or something, that they must have had that. Yeah.
But why do PCR?
So this is another thing that strikes me, because like if you assume that this was caused by SARS-1, then it would have spread everywhere, right? Like the people were already traveling back then. And also, where did it go? Like if we start testing for SARS-1 now, would we find it in the people? Probably, or maybe not. Maybe it disappeared.
You cannot make money on anything, so you still make money on tests.
I agree with that. Like I'm just trying to really point out that. No, I think we agree on the overall theme, which is it's a total abuse of the technology. I'm still trying to go really deep into the details and trying to see, where the actual problems in the fine print are. But yeah.
No, the actual problems were that a person that was diagnosed has to be killed, because we will get money. We doctors at the hospital will get money. So what they were doing, they were isolating the patients, giving remdesivir, because this was the Gilead Deep Pockets, a company that produces the most commonly used HIV/AIDS medication. So the remdesivir, although we know that remdesivir is toxic and simply has very high nephrotoxicity, and of course we connect people to attach people to respirators and now, or ventilators here, and because people do not like to have things in their throat and something like that, so we give them medication that will relax their muscles, Pavlon and other medication. We'll tie them to the bed and they die.
Yes, and of course you have some small fraction or certain fraction that doesn't do well, and they pass.
Yeah, but then
faster than normal, I guess.
Yeah. And then, as you know, the statistics, people who die of COVID, all were not vaccinated. That's the statistics.
Well,
and the CDC, when Ms. Valencia was asked what's the percentage of people that died, she said, no, we don't keep the statistics.
Yes, exactly. Yeah, I did a FOIA request. But surprisingly, they can still publish the Our World in Data chart, which is a fraudulent chart, because they include people that they can't verify the vaccination status as unvaccinated. So it's ridiculous. I mean, it's besides that, you can probably not even compare the two groups because they're so confounded. But like it's basic statistical fraud of like how you count. You don't even count properly. And I don't know if they publish that chart or if that's Our World in Data that publishes that chart. I've tried to reach out to the founder or the person, the main scientist at Our World in Data, and he just blocked me straight up, because apparently he's more interested in getting the money from Bill Gates and other people than doing proper science. But yeah, I mean, I have another interesting thing to share. Let me see. So I did. Can you see my? Okay, yeah, I have my window shared now, I guess. So I reached out to Edward Holmes, who's the I think the only Western. Yeah, he's a virologist from Australia. I reached out to him because he's the only Western person on the Bou et al. Paper, I guess. I actually reached out to all of the authors, but he was the only one who initially replied. And I asked him about, because we did find some human DNA match in the beginning, and they were aware of that too. So we did get three versions of they have 3 versions of the genome. So they have this is in GenBank, the MN 47.1.2.3 like version 1, version 2, and version 3, and they did remove the human genome reads, as I guess in the, I believe it was, it's been a while since I did this. I think they removed it, I think, after the first one. So the first version contains the human genome. I mean, the human genome matches some of the human genome. Let's just rephrase it. Some of the reads that match against the human genome. In version 2, I believe they updated it to the in version 3, from 2 to 3, they updated it to the race. So they did a race amplification of the ends to find the true ends, and then they updated it. So it's ATTA is the final version. They also added the poly-A tail in the version 3 here. But yeah, so like he did confirm that to me via email, while he didn't have any bioinformatics knowledge, I guess, and he said I should ask the Mr. Zhang from China. He does acknowledge that the initially uploaded version had matches to the human DNA at the 5 and 3 ends, which was then corrected. So I asked them if they can publish the data, all the raw data, but they can't, of course, because, well, they say ethical problems. But so I mean, because they established this sequence, right, with the next generation sequencing with the short reads. So they actually proved the point that they can get some human reads in there, into their final sequence.
Yeah, but that's very easy. That's very easy.
Problem?
That's very easy if they didn't, then search for human. If they assembled this virus from a, Illumina type short read sequencer, NGS, then of course, if they use as a master sequence, a sequence that already had some human DNA, or, this mammal DNA, then they of course assembled it with non-viral nucleotides. So that's very simple. And they are, let's say, when I was a gene bank curator for maybe something like 8 years in the 90s, and a lot of sequences that were submitted without next generation sequencing were contaminated with plasmid sequences on which somebody cloned something. So people that didn't have any bioinformatic tools were unable to remove the sequence that shouldn't be there. Yeah. So this is very common from the times, even from before the next generation sequencing and assembling to a master sequence. So this is nothing unusual, but it doesn't really matter if the primers were not really for diagnosis, were not really developed on this sequence, yeah, on sequence that was not viral sequence. But even if they were on the viral sequence, you don't know really what you breed, the billions of viruses that you get every day. So cross amplification is possible. And that's why PCR should not be used for diagnosis. And this is what they say, what they said, all kind of big experts, that PCR is a gold standard in sequencing. And this is not. The EPIC papers published 2015 say exactly, do not do PCR on swabs for respiratory infections. These are very important papers that everybody should know, especially pulmonologists.
Yeah, well, I mean, so you all have obviously you have those problems with the detection, with the amplification of the tests. But I think there is a possibility that there is still also a problem with the sequence, that the target sequence that we're looking for, which is the SARS-2 sequence, what they say. But I mean, I think there are two papers, as I said, where they said, I think there's one from China CDC and there's one from, I think, South America or I think it was South America, where they did like the Sanger sequencing, which is very long sequencing. But I think for both of them, they have not published the actual raw data. So this is very interesting. But the problem, in my opinion, is like, how do you know that the reads you're getting with the short read sequencing are really from the virus and not from some other bacteria or human?
Yeah, because of overlaps.
Contamination.
Because of overlaps. That means, you have the coverage of a sequence from run on a NGS instrument. You have multiple coverage. So that means only things that are overlapping.
Yes, it's the probability. Yes.
Yeah. So this is not a big deal. And then, of course, you have to be good in all kind of bioinformatic tools, which exist, and some are free. So people sometimes do mistakes. And interestingly, as I think Holmes explained, that the sequencing was not done in Wuhan, was done in Shanghai.
By not in my email.
By somebody
yes.
who
maybe, yes.
Yeah, and he told also the story that they took from Wuhan to Shanghai the sample on a train. And this, he was, of course, pretending that that's a very bad virus and that if you have it on the train, you can infect the whole train. But he's unfortunately not too bright, because how come if the virus was on the Diamond Princess cruise ship, on the Theodore Roosevelt aircraft carrier and the Gulf aircraft carrier, why people survived?
Yes.
If this is a deadly virus. So unfortunately, Holmes is not too intelligent, because he should understand he cannot push the theory that this was a deadly virus when there are clear signs that it was not a deadly virus. But remember, they were pushing the story of this Chinese ophthalmologist who just saw a patient who had the Wuhan virus and died at 32 years of age. So remember that he had his obituary is on Wikipedia and in Lancet. How far this lie went, huh?
Yeah.
But paid by Pfizer, so.
Yeah. Do you think the human genome? What do you know about the Human Genome Project, and do you think the what we know about the human genome is accurate, or is it? What are your thoughts?
The thing is that in 2004, not 2004, in 2009, when Obama became president, there was this project, because the cost of sequencing DNA went down a lot. So theoretically, it went down to less than $1,000 per genome. And there was a project which was called this personalized genomics, which made a lot of sense because there was a very good database of DNA. That means it was the NHANES project that CDC was running, I think, since the 70s or maybe even earlier. There were volunteers, there were several thousand volunteers that would sign up, and I think once per year, their blood would be collected and their health history. Of course, this was detached from, their personal identification. So this was a very good database, and I was part of a committee that was meeting together with NIH, And this was Francis Collins, who was for a long time a director of the NIH. And everything was set in stone at some point. The money was almost there from NIH. The CDC could use these enhanced samples. Of course, CDC would not do the sequencing because CDC didn't have such big labs. But Stanford University was doing the sequencing of HIV on a large scale for the CDC. So they thought that David Relman's lab at Stanford could do this project. I don't know how many samples per year they wanted to do, but in the hundreds. So it was not in the thousands, but in the hundreds because of the money available. And then somebody from Obama's circle pulled the plug on that because they said that this will make people whose DNA or genes will be known vulnerable to all kinds of stuff like blackmail and whatever. So because of personal information, which was not there at the sample, but they said, okay, they will determine that a piece of DNA like that means that you are going to be a mass murderer or something like that. So Obama said, no, it cannot be done because it will violate people's privacy. But collecting, let's say, I don't know, some other information is not considered that, but they then, and I thought it was a great project because you could find, how people should be protected for certain, let's say, even cancers or something like that. But unfortunately, it was not done. So now, which is like, 2009, 2025, we are 16 years later, but still nobody is talking about this project. But the Human Genome Project is, nobody really questioned, that this was something that was not done properly. What is being done on a much smaller scale is, of course, if somebody has cancer, the cancer is the cancerous cells are taken and the complete genome is done. And for this reason, for instance, for breast cancer, it's known that if there are 50 mutations, then it becomes very, very bad type of cancer. So many things are done on cancers, and nobody really says that this violates people's privacy.
Yeah, I do wonder though if it's maybe still possible that this is a whole sort of like a misunderstanding even. Like I think it's because I mean the human genome, what they say, like I can download the human genome right now. I can go to the website and they have it like 3.1 gigabyte, I think is the human genome. That's about what the Windows 7 image has or something. So that's one interesting point to believe that a human genome is just about like a CD or a DVD of Windows 7. So my question is it really what we know as a human genome? Is that really the full depth of what makes up a human? Can we essentially recreate a human? Could we, if we had the technology, the tools, we could just take that code and recreate a human, that person that was.
No, the old, I don't know when it was formed. The saying, you can create a cell only from a cell.
Yeah.
So that means if you have the DNA sequence, it's very difficult. You have to have a cell and replace the nucleus, and then you have to pray that no mutations were there that are fatal or lethal, and that this cell can then give a rise to.
So you have a lot of organs. So technically, it's not possible right now. I believe I've looked up. I think there are attempts to do it with small bacteria, maybe, but I don't know. I don't think it has been really done. But I would go further. I would say, are these information? It's just information in the end, right? It's a sequence of 4 nucleotides. Is this information really what makes up the life, right? Is this really
You forget.
Or are we looking at, like, because like an analogy I could make is that we could take a lot of hair samples from people and we could look at the hair samples. We could look at, okay, brown, curly, whatever. We could probably make out where the people are from roughly, or what race they are or something. So isn't it sort of like the same similar thing that we're looking at very high resolution signals, and we're trying to make some sense out of it, but it doesn't necessarily mean that these signals represent the code that makes us.
Look,
would you? Yeah,
we can reduce everything. Do you know that people are studying the flu virus for over 100 years, and we don't know what the flu virus genes really do?
Yeah.
So sequence is not enough. Sequence,
yes.
Cannot be tested. Even genes. You can now slowly decode what kind of information is read, because you have all the problems. How the information is read. You have the introns, the exons. You have different starts for transcription. You have some post-translational modifications. So it's even the primary information is very difficult to get hold of. But even for viruses, what are those genes really doing? And the same with this. They say, oh, these few nucleotides change in the fiber gene of coronavirus means something. Remember the people that say, oh, Wuhan lab or Barik or whoever, Dashak or whoever else created the furin site in the fiber protein of the coronavirus, the Wuhan virus, and then this virus can infect humans. How stupid. The whole virus is 30,000 nucleotides. The fiber protein is just about 1,000 amino acids, 3,000 nucleotides. So the change in one gene will now enable this virus that was from some different organism, to compensate for all the changes that are needed, so this virus will infect humans. Look, for instance, Holmes, I think, described some viruses from bats that are much closer to the Wuhan virus than, of course, the SARS-1 virus. But still, there are a lot of changes to make this virus, possible to grow on human cells. And the furin, these people are so crazy, and some of them, I think, are crazy on purpose, because they are molecular biologists, and they should know that this is not enough. That adaptation of a virus from one organism to another organism requires a lot of changes, because the proteins are different. So that's a big problem. But we are dealing with fraud. We are dealing with fake science. We are dealing with stupid people that now are people that look at Kevin McKernan and his theories. He now found this promoter, but people do not understand who promoter is, of the SV40 virus, which is not a virus that causes cancer in humans. Nobody proved that.
There's no way to prove that. How would you prove it? Because you would have to look at 1,000 people.
You would have to infect them. You would have to infect them.
And you have to do that, or exactly.
So they just did it on cells, but never they found this virus in humans. So it's not a cancerous. That doesn't cause human cancer.
And just to find it in a cancer, it doesn't mean anything. Like you would have to. I told him, and that's where, I guess he blocked me before already. But I told him, don't you have to look at like 100 people with cancer that are vaccinated and 100 people that are unvaccinated, and see if you find a significant difference. If you can only find it in the people vaccinated, I mean, it's things like that. And it's
he's a heavy user of drugs, and he boasted about that, and he switched from dope, from marijuana to this mushroom dust. So he and then everybody believes him. Because he said
that's really the.
They do not understand that this plasmid was not added to the vaccine, and it's just a minuscule amount that was not removed with DNA. So You know,
Well,
okay.
It's a whole distraction because yeah, we can end it up, but in my opinion, it's a whole distraction because I mean, even if you remove it, besides it being a problem or not, even if you remove it, like you still have the transfection there, and you would have to ask, okay, what is the purpose of the transfection to start with? Like where is it? Where is the efficacy? You know, I mean, like.
Yeah, but you cannot remove it completely because no biological reaction has zero endpoint.
Yeah.
Okay, it's good to,
Interesting.
to finish now.
Yeah.
We can meet next week or another time, you know, but there are a lot of topics that we can discuss.
I think as you said, the core problem is that it's so complex, the whole topic, and there's so many unknowns, even for microbiologists and geneticists, that we really don't know. We've only touched the surface of it. That's my understanding, and so and people believe what people like McKernan just write on Twitter, and then the belief is on both sides. It's on the other side as well, and people don't seem to question because they don't understand it. That's the real problem, I think. Yeah. Well, anyway, thank you very much for your time.
Thank you, man.
See you, next week or in maybe 2 weeks or so. Yeah, thank you.
Yeah, we'll stay in touch.
Yes.
Okay, bye.